So it gives me great pleasure to introduce our next speaker, Dr. Douglas Fraser, professor, clinician scientist at Western University in London. He's going to talk about blood biomarkers and mild traumatic brain injury. He is a fellow of the Royal College of Physicians and Surgeons of Canada and co-founder and chief medical officer of Neurolytics, a diagnostics-based biotech company. His research is focused on identifying diagnostics, prognostic biomarkers for acquired brain injuries. Welcome.
Thanks so much for the invitation. It's good to see everybody again. I'm going to just start off actually with a little bit of history on the biomarkers. I've been working on biomarkers since 2008. And in fact, we did one of the first studies with GFAP. At that point, there were actually no commercially available diagnostic tests, except for a company out of Germany that was using a GFAP ELISA to test to see if people were feeding animals central nervous system. So they would go around with agricultural government agencies and test feed. And that was the actual kit we started off with. So it just tells you how much things have changed over time. So everybody knows how we categorize mild traumatic brain injury. We still use the Glasgow Coma scale. It does serve a purpose. We're all talking the same language. We pretty much all do it the same way. It's great for communication, but it is extremely limited. Two populations that I'm particularly interested in. I am a pediatric critical care medicine physician, so I am interested in the pediatric population, and I'm glad Chris talked so much about kids. But I also got really interested in the military because I believe they're a vulnerable population that needs protection as well. In general, the type of mild traumatic brain injury the two groups are facing are different, with kids, of course, having the mechanical injury and the military facing blast injury or pressure wave changes. And that's largely due to their training. It's not due to being in theater the vast majority of time. So again, the populations I'm interested in. For a biomarker to be valuable, let's start really at a basic level. It needs to be sensitive, it needs to be specific, and it needs to be reproducible. It needs to be affordable and widely available. It needs to be standardized across institutions. And the most important thing is it must be linked to a clinical decision. There's not much point in measuring all these things, spending lots of money, unless it's going to change something you're going to do clinically. It could be diagnostic. Do they have the injury? And that's what I'll be primarily speaking about today. It can also be used to avoid other harmful tests such as CT scans. And a great example is that many biomarkers right now are being used to determine whether adults need CT scans or not. In the pediatric world, it would be disastrous because they're just not specific enough. And in kids, it would actually result in more CT scans, and a single CT scan on a child will increase their lifetime risk of intracranial cancer. Prognostics, how will they do after an injury? Will it aid advanced planning? So resource mobilization, speaking with families, speaking with patients, all those types of things, stratifying for studies. Will it help with therapeutics and rehabilitation? And will it allow us to inform people when they can go back to their regular activities? You just saw a slide very similar to this. And again, it's just telling you about the basic biomarkers that we use. There are more coming out all the time. But in general, it's the astrocyte markers that have been used most and seem to hold the most promise. That includes the S100 beta, which is a calcium binding protein, and GFAP, which is an intermediate filament that's involved in the structural aspect of glial cells. And then in the neuronal side, you have NSE, you have CHL1, and along the axons, you've got NFL and tau, which you just heard about. We don't understand really how these things are released. After 20 years of research, we don't really have a good idea. Cells are not always dying after an injury and spitting these things out. So how are they getting out, and so that we can measure them? One example with GFAP, we use human astrocytes in our laboratory, and when you concuss a human astrocyte, the end feet, which cover the majority of the capillaries and arterioles in the central nervous system, right after they're injured, will slowly retract off the blood vessel. So you actually get metabolic uncoupling after a concussion related to the astrocyte. And then over the next hours and days, those end feet start to grow back and find their spot again. That process involves a lot of remodeling of the GFAP, which is a structural intermediate filament, and that may be involved in the production and release of GFAP in particular. Another thing we don't understand is how these things get in the blood. We talk all the time that it's breakdown of the blood-brain barrier, but that sounds a little simplistic, and it doesn't always occur. But it may be one component. We also don't know if these are being transported through the CSF and to the arachnoid granulations, they're being taken up into the venous system, or with some new theories with the lymphatic system during slow wave sleep, you have these waves of CSF that go across your brain. They may be picking up these proteins or toxins, moving them down to the perivascular space, and then they're getting somehow into the blood from that standpoint. But we really don't understand these things. Because we don't understand these things, it's really hard to get an idea about the kinetics of a plasma biomarker related to blood. Just a little bit more information. These are the common ones which we've seen over and over again. Again, GFAP probably being used the most. That's the one we started with over 16 years ago. Astrocytes and neurons tend to be the primary location, but there are other sources. So again, if you're measuring them in plasma, you have to be concerned with this. GFAP, we have found it after abdominal trauma in kids because it is around the pancreas. So you have to be aware of these things. The half-life is different, as well as the peak. So when they actually reach their peak concentration in plasma. And because of all these things, and because we don't understand the kinetics, it becomes very, very difficult to design studies and to do good studies. This is just a graphic, again, showing how these things go up, what we think is happening. Unfortunately, this is not just for MTBI. This is for TBI and all sorts of neurological injuries. These have been, the data has been put together. But in general, most of them come up reasonably quickly, with the exception of NFL, which you just heard about, which goes up for a long period of time and stays up. So how are these actually being used? And, two guidelines which have come out not so long ago. There's a Scandinavian guideline, which utilizes S100 beta, but you have to measure it under 6 hours. And it's only in a low-risk population that they would use it. And they would measure it, and if it's negative, they would send you home. If it's positive above a certain threshold, you go for a CT scan. If the CT scan's normal, you would go home. If the CT scan's positive, you would come into hospital for observation. So this is one example based on a guideline on how a biomarker is currently being used. Another example comes from the French guidelines, where they've expanded that time frame. 6 hours wasn't enough, so they went to using S100 beta under three hours and using GFAP under 12 hours. This is only for the medium risk population, but that medium risk population is quite conservative in the sense that you have to be over the age of 65, or you have to be on antiplatelet drugs and so forth. But regardless, these two examples have theoretically reduced the number of CT scans by about 30% in the emergency room. At the end of the day, they're giving you something objective to go on, as opposed to our CT rules, which we commonly use, which are clinically based in the ER. They're very good also, but physicians tend to do them, and then they order a CT anyway because they're so worried about missing something. So now you've got something objective. But at the end of the day, and I don't mean to sound pessimistic, but after 20 years of research, after hundreds of millions of dollars, after so much effort and time, we really haven't gotten very far with blood biomarkers. So now I'm going to switch up and I'm going to start showing you some data. And this data is from the SHRED program out of Calgary, who's really done a fabulous job. Jason Tabor, who's their PhD student, has really run with this. And Cheryl Wellington's lab's done a great job as a clinical chemist. They've been very, very careful how they approach this. And I'm fortunate to collaborate with them. This was a paper that was published in Journal of Neurotrauma last year. And they looked at kids. They went right across the country. There's a whole bunch of sites. They looked at about 1,000 kids they had baselines, plus they had kids who have been concussed. I can tell you that the kids, even though it's a cohort study, a significant number of those kids, they had a baseline and an injury in the same child. And the cohort study versus the single subject study actually replicated each other quite well, which is good news if you're doing cohort work. But what I want to point out here, these are your common biomarkers. These were done with this Quanterix Simoa platform in Cheryl Wellington's lab. And it's a very sensitive tool, a very sensitive platform. And they took time to really look at baseline characteristics, which is so important. And I think a lot of us are guilty of not really putting in the time to do this. But in this particular case, you've got no history of concussion, a history of concussion. First thing I want to point out to you on this is that there's really no difference using these traditional markers in kids who have had a past history of concussion versus those that haven't. That's the first thing I want to point out. And I'm going to come back to that later on in the talk. But the second thing I want to point out is even at a baseline level, there's really quite a bit of variability in these particular markers. If you take total tau, the very first one, the median value is about 1.8, but you've got markers that are going up to 3.8 and down to around 0.5. So there's a tremendous range even at a baseline level. So then you think when you're injured, you've got patient heterogeneity and you've got tremendous injury heterogeneity. And I'll just, as a critical care specialist, when I care for severe traumatic brain injury, I was always surprised when you look at a CT scan on admission and you would just see some global edema, some white gray matter differentiation issues, so forth. But then you would do an MRI at 72 hours and you would see very focal, patchy events. And it was different for every child. So clearly there's amazing injury heterogeneity that's occurring with all of these injuries. So when you think of that, all of the studies out there that have positive results, and we all do them, we use population statistics. And those population statistics are really good for giving us a p-value and saying the population as a whole is different, but it really doesn't tell you much about an individual. And that's another problem we have with these studies. This was done very nicely. That was one example. I'll show you one other example. Again, this is from Jason's paper, this Calgary Shred group. And what they did is they looked at kids over time and they looked at the baseline levels of these markers. And all the markers were fairly consistent with the exception of GFAP. We've shown this previously, other people have shown this previously, but over time, GFAP actually decreases at a baseline level. So it makes it really, really difficult to even come up with a cutoff value when it's constantly changing with development. And then again, if you take tau in that first one, just look at the spread of the values at a baseline level. Again, lots of patient, lots of injury heterogeneity. Now, they were kind enough to give us the data on over 1,000 kids. In my lab, we've got a biorepository. We're just creating a big proteomics lab, which has been funded. And then we've got machine learning and bioinformatics. And my machine learning group went after this data to try to get some idea of how it could be valuable. And in particular, we want to know about the individual. And if one biomarker is not good enough, what happens when we combine all these biomarkers? So this is our first kick at the can. It's not been published yet. It will be published soon. This is only the biomarker data. We've looked at everything, the scats, the histories, everything. I can tell you the scat gives an area under the curve of about 0.75. And as you know, areas under the curve greater than 0.7 are good, greater than 0.8 are very good, greater than 0.9 are excellent in terms of diagnostic tools. So the scat comes in 0.75, which is good. The blood biomarkers, these four, when we look at a large number of kids, have an AUC of only 0.62. So these essentially are not really all that valuable. And that's an optimized model, okay? If you look on the right-hand side, you've got a UMAP, which is dimensionality reduction technique. And it does look like there's some organization there, but it's not good enough from a diagnostic perspective to really help out. If you go down to the left corner, we do feature importance to try to determine which of these is giving the best information. And GFAP always comes out ahead as being the best biomarker of the group. But even when you combine GFAP with the other biomarkers, it really doesn't get much better. And in the bottom right, you've got a SHAP diagram, which is related to our decision trees. And it's essentially telling us, you see the red off to the far right with GFAP. And again, that's giving you some information. That's a reasonably good biomarker. UCLH, maybe a little bit after that. The NFL and the t-tau really aren't helpful at all. So this is a slide that I created probably about 15 years ago, but I think it still holds true. When I started off on the left-hand side, we had these tests which really gave us a good specificity. They were your standard ELISA tests and okay specificity. And then we've started to build these ultra-sensitive protein assay kits, which are great. So you've got the Simoa, you've got other ones as well. And what we've done, though, is I think we brought up the background as much as we brought up the signal. So yes, they're very, very sensitive. You're unlikely to miss people, but you're also likely to bring up unnecessary or patients that aren't actually positive. again, a goal of what we do, given that we want this work to correlate with symptom severity or outcomes, something that's clinically relevant. And when we look at one concentration of a biomarker, if we use a typical conventional statistic, if we do a correlation, you can see a really great correlation there. But if you look actually at the concentration of that one biomarker, what does it really mean for the symptom severity? So that comes back to the fact that one biomarker, again, is unlikely to really help us in this problem. So we have to go to a biomarker panel where we start to see something like this. And again, we've been talking about this for 15 years. We're still not there yet, but I hope to get there soon. So I'm going to switch it up now and talk about some work we're doing in our lab for this last little bit. We do a lot of omics, whether it be proteomics, metabolomics, and now we're fortunate to have everything under one roof. So we collect blood samples. In some cases, we've done CSF as well. And then we have opportunities to do conventional statistics, but we also have an excellent machine learning group. And now we have all the bioinformatics analyses. I'm just going to show you little snippets that I think are important for today. We did a study on males 13 to 14 years from bantam hockey. And as Chris said, yes, they hit. The real issue with these kids, though, is some of them are really, really small. They haven't even gone through puberty yet. And then you've got a kid who's 6'2", and they're playing against each other and they're hitting. So these kids are really having some fairly dramatic concussions. And then we take that work, we go to the proteomics world, which I won't talk too much about, but we can get into precision medicine. We can determine the cell types that are involved, signaling pathways, and eventually get to diagnostic biomarkers and even drug therapies through drug repurposing. So here's an example. In the top, you've got control kids that have not had a concussion, no history of concussion, no acute concussion, but they played hockey. Everything else was matched, age, sex, sport. In the middle, you've got the acutely concussed kids. And at the bottom, you've got a group of kids who have not had a concussion in the past 6 months, but they have a history of concussions. So they could be 2 years, 3 years, 4 years out. It could be anywhere from 1 to 4 concussions. And if you look at that data, red means a protein has gone up, blue means a protein's gone down. So it's a relative expression. When we start to work with it a little bit, so if we take the mean values of all those proteins in the middle, you see the controls on the top, you see the acute concussion, that's a very different pattern. And then at the bottom, you've got another pattern again. When we do hierarchical clustering, so we start to group these proteins down into organizational patterns like cytokines, chemokines, that type of thing, you see these 3 groups are extremely different. And what really jumps out at me, I'm not surprised about the acute concussion, but really concerns me and Chris as well, I'm sure, is that those kids who've not had a concussion in the past 6 months, they could be years out of their concussions. They have a signature, and that signature is very, very different than a control, which tells you, yes, the SCAT may have normalized. Yes, they might be back in school. Yes, they might be back playing sport. They are not biologically normal. And it's absolutely clear with this work. So it's a very, very important point, I think. You can see using a number of different techniques that the groups are different. I won't get into all these, but we can look at the cell types that are involved. And in the acute concussions, it seems to be a CD4 T cell activated event involved with inflammation. And then in those chronic kids, they actually have mast cells that remain activated. And this tells us something because now we can start to look for biomarkers that are related to those cell types, but also therapies. We can start to get into the signaling pathways, and we find that there's vascular endothelial changes, there's extracellular matrix changes, but we can start to identify the individual proteins and then rationally go back and look to see, are these good biomarkers of injury or not? In this particular case, now the majority of proteins actually go down in concussion, which is very different. We've done lots of work on COVID, sepsis, that type of thing. The majority of proteins go up, about 75%. With concussion, the majority actually go down. But some still go up, and that pathway seems to be largely regulated by IL-8, which seems to be that centerpiece, and then it's modulating a whole bunch of pathways around it. So there are some definite changes going on here and some definite biomarkers. One could argue maybe not all these markers are specific for the central nervous system, so you have to put them into the correct context, but they may be helpful when we mix these with other symptoms. We also use machine learning. We use a number of different techniques to bring down the number of proteins. So when you're measuring 3,000 proteins, if you use conventional statistics, you have to control for multiple comparisons. If you use a Bonferroni or something like that, at the end of the day, you're almost left with no data because it's too conservative of a test. You can use some other less conservative tests, but at the end of the day, we found that if we use feature importance and we narrow down the important number, and then we go into our conventional statistics, we're much more successful. Here are some proteins that we've discovered from adolescent concussion. The majority go down. They're all expressed in brain in high levels, particularly ATOX1, which is in the cerebellum. You've got SPARC, which is in brain tissue, brain capillaries, and then you've got an adenosine enzyme, which is also expressed in brain. So there are three new biomarkers. What we then do is try to bring them together to see again how they work. And if you go to the bottom right, you see that we get a much better AUC if we bring the three together, if they reach a certain threshold after concussion. In saying this data looks so good, it's so exciting, but my experience, I'm sure your experience also, as soon as you go to validate this in external population, you're usually off by quite a bit. This is a very tight population we're looking at, and it doesn't always hold true when you go generalization. We can show these things with a number of different techniques. And with my last few seconds, I'm also just going to show some very brief stuff on the military. So these are blast injured individuals. They also have a proteome, which is completely different between those who have been exposed to blast and those that haven't. We can use, again, machine learning to start narrowing down the different proteins and coming up with our AUCs. And we can put together in combinations. And we find that a combination of two actually works quite well for blast injury, being LAIR2 and PTGR1. And those separate out the patients quite nicely. So these are the approaches we're using now. I think we need to move beyond the traditional biomarkers. I just don't think they're giving us enough data. I think we have to start looking at combinations, and that's what we're after. That's what we're doing. And my last slide, we also, if we're going to test for people, we need to come up with something which is easy, something which is readily accessible. So we're working with a lot of lateral flow companies now, and we're designing lateral flow assays where you can measure more than one protein. In this particular case, you see a lateral flow cassette in the bottom, which has room for 3 proteins. You can do a capillary pick on your finger. You can suck up 40 microliter. All the conjugates are in that little bottle. Drop down some drops. They find their way out. In this particular case, we use nanodots, which fluoresce, and you need that little fluorescent flashlight. And those are the types of tests we're trying to develop as well. Sorry, I went over by a minute. Thank you.
Thank you. I guess we'll start with somebody online who asks, in children, are non-injured biomarkers different at different ages? And if so, how do you then compare to injured children of different ages?
So absolutely, the GFAP is the classic one. It goes down with time. And this is why I think the SHRED program's done such a good job, is they took the time to look at the baseline characteristics to see what you're starting with to begin with. What we do in all of our studies is everyone is age, sex, and activity matched. So hopefully we're working within a very tight range. But if you want to expand these out, yes, you have to do that. We also do metabolomics, and guess what? The metabolomics change between sex and with development, depending which metabolite you're looking at. So you have to take these things very seriously, and you have to look at each one individually.
Yeah, no, thank you. Are there any other questions?
You mentioned that use of biomarkers in the ER reduced the CT scans, but how widely available are they in ERs?
Yeah, so I'm not an adult emerge physician. This was data from Scandinavia and France. To the best of my knowledge, we're not using them in Canada or throughout most centers in the US. If I'm wrong, please someone correct me. One of the problems, again, is standardizing and cost. And I think there's still quite a bit of doubt as to how helpful they are in the overall spectrum of things.
Hi. Thank you. That's great. Great research, and I'm really hopeful for your success going forward. And I'm wondering, I really like the new approaches that you've been taking. I'm wondering, are you going to introduce baseline testing? I know that's kind of gone out of favor in terms of behavioral testing, but are you interested in that for your work?
So in the beginning, absolutely. So one of the large scale studies we're doing right now with metabolomics, is every child that is in the centers that are doing it, at the beginning of the sports season, they have to do a full C.E.T., physical exam, big history, but then they also give a blood, a capillary sample for us, and we bank that. And then if they get injured, we've got something to compare to. So absolutely, you have to do that in the beginning. Hopefully the day comes though where we've got clear idea of what those cutoffs are and what the normal range is, and we don't have to do that anymore.
So I was wondering if you do that, I'm happy to hear that. Is there any way to standardize some of the factors that are transient, like a person's hydration? Could you, for example, just make sure that all kids are hydrated at baseline, and then if they get a concussion, that would be something that would be innocuously mimicable, I would imagine.
Yeah, fabulous questions. And the short answer is so important to keep an eye on those factors and to try to record them as best as possible. But to try and control for those in a sporting event, in a study, extremely difficult. And I guess you could also argue that if you have to do that, then maybe the biomarkers aren't that good because that's not real life.
Was one question from our machine learning scientist who wanted to know which specific machine learning techniques you were employing in your research.
Oh my goodness. So everything from cross-validation, dimensionality reduction, random forests, a wide variety. We have 3 machine learning people right now, and they all have their own interests. What's nice is when we look at the data, is we use multiple techniques to make sure that the results are consistent. And then we'll also go back when possible and compare them to conventional statistics to, again, make sure we've got consistency.
Great. Thank you so much.